They're stuck. And if you're awake, you realize that the acronym for that is SOS. So take that as you will.
Okay, thanks, Hans. First of all, I'd like to thank the organisers for inviting me here. And when I asked what I should speak about, and so they said to give more a sort of tutorial type talk on the background of some work particularly for the students and new people coming into the field. So as I mentioned last week, originally my idea was to talk about the Turing model and then about dictyostelium, but then that's already been covered. So then that's why I thought, well, I'll just talk about some other stuff. And this really is aimed at the graduate students. And when Anatita told me that there were sort of people coming into the field that were new, I didn't realize that literally a lot of you had started your doctorate like last week or the week before. So this is very much aimed at you, so please do ask questions and please engage in the talk. So I thought I would talk about three things. So one thing I talk about my sort of personal view of the role of modeling. So I thought it's Monday morning, so everybody's a bit tired, so then this would perk everybody up and get them annoyed, and then they would start getting very passionate. And then I'm going to talk about two applications, this application here, which is the same thing, and then neural crest cell. So a lot of the stuff I'm going to talk about is quite old stuff, and a lot of it isn't mine. This stuff here is stuff we're working on at the moment, but mainly it's to try and give you a sense of the role of modeling and what a model can do and what it can't do. So we've heard a lot about modeling. We haven't really had much discussion about what a model is. And a model is a different thing to different people. Okay? If you ask the person in the street what's a model, they would probably say one of these three things. Okay? So if you talk to a biologist, and when a biologist refers to the model, they mean a mouse or a chick. If they're doing in vivo, or in vitro, they mean a Petri dish with some cells in the Petri dish. And if you work in things like cancer growth or something like that, so this has gone off again, so I'll use the big stick, then you're making the assumption that what happens in the mouse and the chick is what happens in the human. And in many cases, that's actually not true. But it's a model. If you talk about cells in the dish, when you put a cell in the dish, you've changed all its mechanical properties. Due to mechanotransduction, you've changed a lot of its biochemistry. So when you do an experiment to discuss the behavior of a cell in a dish, what you find out is how a cell behaves in a dish. Okay? And then there's equations. You've seen a lot of equations last week. There are computer models, rule-based models, statistical models. So when a statistician talks about a model, they mean something completely different to when a mathematician talks about a model. So all these are different definitions of model. They all have one thing in common, which is they're all wrong. Okay? But some are useful, to paraphrase statistician George Box. And that's the key thing. Some are useful. Okay? So to give, and this is, some of you will know this, Lewis Carroll, famous mathematician, paradox of the complete map. So here's a discussion about map making, and this person here is saying, we've carried, that's another thing we've learned from your nation, map making, but we've carried it much further than you. What do you consider the largest map that would be really useful? 6 inches to the mile. Only 6 inches. We very soon got to 6 yards to the mile. Then we tried 100 yards to the mile. And then came the grandest idea of all. We actually made a map of the country on the scale of a mile to the mile. Have you used it much? It's never been spread out yet. The farmers objected. They said it would cover the whole country and shut out the sunlight. So now we use the country itself as its own map, and I assure you it does nearly as well. Okay, so we're not, in modeling, we're not trying to do mile to a mile. Because if we did a mile to a mile, it would be useless. And if you're going to do a mile to a mile, why not use the country itself? We're trying to do 6 inches to the mile, abstraction, because that we can use. And then Turing, who developed the reaction diffusion models you heard about last week, this was his definition in his paper. So this model will be a simplification and an idealization and consequently a falsification. So again, it's wrong. It is to be hoped that the features retained for discussion are those of greatest importance in the present state of knowledge. So to me, when he says hoped, suggests to me that this is not a science, it's an art. Retain the feature of greatest importance. Now, if you're a modeler, the chances are you're going to describe a situation and you're going to ignore something that someone in the audience has spent their whole life working on. So one thing the new people in the audience, new students need to realize, you need to develop a thick skin. So when people shout at you, you don't start crying, which is what I used to do, but I don't do anymore. And now I just drink. So another thing is important to note is that if your model is good, it should change the present state of knowledge. And if it changes the present state of knowledge, then you have to start all over again and maybe come up with a different model. And this is precisely what Hans was saying last week when he quoted John Bonner about this rocking back and forth between theory and experiment. And in the British grant system, when you talk about this, you talk about the virtuous circle: predict, test, refine, predict. Okay, so you generate hypotheses, you make predictions, you test them experimentally. Some of them agree with the experiment, some don't agree with the experiment. You therefore refine your model and you start all over again, predict, etc. And Leah Edelstein, Keshet pointed out last week, she said that sometimes you learn more when the model is wrong. And I would agree with that, because if what you're doing with your model is testing hypotheses, what you're doing is you're taking what the biologists think is happening and you compute the result of that, and you find that that's not actually what happens. It means there's something else to look for. And that can be very instructive. And here are just a couple of really nice articles that you might want to read concerning modeling. So this is a paper by Jeremy Gunawardena. So I realize you probably wouldn't be able to read his name, so I've written the name down here. So Jeremy is actually a pure mathematician by training, now works at Harvard Medical School, runs his own lab, does experiments. And this is in BMC Biology 2014. It's entitled Models in Biology, Accurate Descriptions of Our Pathetic Thinking. Okay, I think that's really important because last week a lot of times people were being asked, speakers were being asked the questions, what about this? Have you included this? Have you done that? Have you done other thing? And the answer in a lot of cases was, no, I don't know, maybe, but I haven't tried that. It's not the modeler's fault, it's as a community. It's our level of understanding. What a modeler is doing is trying to put into a formal setting our level of understanding. And then there's a very nice paper, Experimental Physiology, 93, page 16 to 26, by my colleague Dennis Noble. And the reason why I put this up is not because of Claude Bernard, but because within this he gives 10 rules, 10 principles of systems biology. And so it would be really worth the new people in the field to read some of the stuff that Dennis Noble has done over the sort of philosophy of modeling. And this is the sort of old style view of how things worked in biology. You had genes coded for proteins, and that gave pathways, et cetera, all to the organism. Very much a feed forward. Everything is determined from down here. But that's not true. What's actually happening is there are feedbacks happening, which makes life a lot more complicated, because feed forward is fine, because that then we can think in a linear way. A upregulates B, B upregulates C, C upregulates D. What happens if I upregulate A? Well, you upregulate D. Verbal modeling, linear way of thinking works perfectly. When you start getting feedbacks, that's when our verbal reasoning doesn't work. And we need some framework in which to compute the outcome of these feed forwards and feedbacks. And that's where mathematical modeling comes in. And notice another thing, which again hasn't been mentioned very much in this talk, in this conference so far, is the scale that's involved. It's a huge scale. And in fact, not only should you go to organism, you should then also go to population and even the environment. There's a huge scale involved here, and that's a huge challenge. The mathematics we use wasn't developed to deal with lots of scales. And so the question is it sufficient for what we're trying to do here? And there was a bit of discussion about that last week, where some people said it was, some people said it wasn't. It's an open question. And this is just a spatial scale. There's also temporal scale involved as well. So those are other things to bear in mind. Now, my personal experience has been the following, and I'm sure a lot of you have had different experiences in this. When I've gone to a biologist and said, you know, I've actually found something out about your system, can you test this? It's never worked. They've never been interested. My collaborations with biologists have worked when the biologists come to me and said, blah, 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 I need a mathematical model. And the first thing I ask then is, what is your question? And the second thing I ask is, why are you asking me? You're a biologist. You've got a question concerning biology. Why don't you answer it? What makes you think I could answer it? In other words, and I think this is the most difficult thing in most research fields, how do you define a question that is amenable in this context to a mathematical analysis? Because not every question is. We all agree that the microscope is really a fantastic thing. You don't use a microscope to measure temperature. You need to first of all figure out, what can I use a microscope for, and you use it for that. And if you can't use it for something else, doesn't mean it's useless. It means you need to use something else. Okay? So what I use mathematical models to do is to test hypotheses, because as I mentioned, verbal reasoning can sometimes be wrong, sometimes it can be right. Biological systems are non-linear, they're not linear. My favorite example of that is if you've got a headache and you take 1 paracetamol, you feel a little bit better. You don't then take 100 paracetamol to feel 100 times better, because biology isn't linear, it's non-linear.
In that case, it's that's what we work on, of course.
Yeah.
And the question is why you can take 100 aspirin or 100 ibuprofen and get an upset stomach, whereas you take 10 paracetamol and you'll die of liver failure.
Yeah. And then when the hypotheses don't work, or the hypotheses are shown not to be sufficient, then generate hypotheses. Also, one might help to prioritize experiments and also to improve our intuition. And so there again, we go back to the Turing model that was mentioned last week. And before the Turing model, if you'd ask somebody, I've got a stabilizing system, and I add to it another stabilizing system, what do I get? would think that most people would say you get an even more stable system. What Turing showed was no, in some cases you get an instability. Stability and stability can lead to instability. That's increased our intuition and led to the idea of short-range activation, long-range inhibition. That's now second nature to us. But it was first brought to our attention by mathematical formalism, a very abstract mathematical formalism.
Sorry, Philip, I love your philosophical introduction. I guess you could put it under the heading of test hypotheses. But one of the things that I guess I have two quick comments. One of them is I would add more specifically for the experimentalists that one of the roles of modeling can be to interpret experiment, because a lot of times you do something experimentally and you don't get the result you expect.
Yeah.
And trying to understand what an experiment means is something that modeling can be very good at.
Yeah.
And the other one comes perhaps under your heading of prioritize experiments, which is we often say that very often you don't need to actually run the model, you just need to build it. Because when you build the model, you identify the missing information.
That's right. Yeah.
You discover that the experimentalist has been focusing on something that by itself can never explain what's going on. And so then you have to say you have to go back and do these additional experiments because you can't analyze the problem that you're trying to analyze.
Yeah.
And so just constructing a formal model by itself, even if you never actually execute that model, can be very important.
I absolutely agree with that, and thanks for pointing that out. That just the act of building the model and the act of having people from a different discipline come at a problem in a different way can bring up issues that you hadn't really thought about. So I'm now going to go on to talk about two examples, and the two examples are going to be examples of invasions, which are going to involve cell motility, or motility of individuals. Now, last week, we heard a lot about what actually goes on inside the cell that generates the ability of the cell to move. And our modeling is going to be, if the cell has got certain properties, how does a group of cells behave? But I'm going to begin not by talking about a cell, but by talking about something that's a bit more cuddly. I don't think I've ever seen one of these creatures. The red squirrel. That was the indigenous squirrel in the UK. And then in the early 1900s, somebody had this great idea to release this guy from America, and released the grey squirrel in actually around this part of the UK. And the grey squirrel has taken over the population now. Okay? That just tells you the Latin name, in case you're interested. I apologize for the quality of this slide. So this is the sort of part of England where we're in at the minute. This is 1964, and it's split up into boxes, and the coloured in boxes are places where people have had reported that they've seen a red squirrel. And then this is 1979, and you can see there are not so many coloured boxes. And one of the nice things is in the olden days, people would write papers. These are quotes, yep.
In terms of the data, do you know how that was achieved?
Yeah, by people saying things like this.
Oh, right, yes.
This is how they collected the data. So somebody would say, because the grey squirrel is a pest, so somebody would say in Scotland Castle, wherever that is, that they cleared his trees of plums in one night, and so then they would say, oh, there's a grey squirrel here. That's essentially...
So that map was done by a literature review then?
Yeah. Or also people just reporting, if they, to conservationists, just saying, we saw a red squirrel outside today in Cambridge, and then they would say that there's a red squirrel there. Here, rat and sparrow clubs offered 6p a head. That's 6, I still call them old pence. That's 2.5 new pence. which actually must have been quite a lot of money around in those days. So here is the simplest model you've seen this week. So G is for gray, and R is for red. And this is a Lotka-Volterra model, and it's the simplest possible model you can have for two species interacting with each other. Okay? So G and R are variables, T is time, everything else is a positive constant. So again, just to explain to the new people, let's assume that there's no R at the minute. Then you've got an equation of the form R1 K minus G times G. So if G is very small, this just looks like RK1G, so it means G increases exponentially. And then as G gets larger and approaches K1, the G by dt goes to 0, and you get a steady state of K1. That's called logistic growth, and it's a very good approximation to lots of species, the growth of bacteria, yeast, etc., some tumor cells, when they're growing by themselves. And there are different types of this model, but basically it's all the same thing, exponential growth and then a tailing off. And all this means is that the growth is hindered by the presence of the other species. Here the red, here the green, here the gray. Okay? Now obviously if you look at these equations and you want to find out how do G and R behave, you want to see, is this positive somewhere because G will increase, or is it negative somewhere so G will decrease? So therefore it makes sense to figure out where is it 0, because then on either side of it either it'll be positive or negative. So let's suppose, so these are straight lines, so suppose when we draw them, so this is G and this is R, and here are those straight lines where the derivatives are 0. Okay? So suppose we're here, which means we've got lots of gray squirrels and very few red squirrels. So we go out with our guns and we kill a lot of the gray squirrels, kill them right down to here, get to here, and then this being in the UK, we then go and have a nice cup of tea. And while we're having a nice cup of tea, the dynamics here, we look and see this is positive, they start to grow, but then there's a switch here, because this is where this was 0, and then this becomes negative, and we go down to here, and we get the red squirrels, and the gray squirrels have disappeared. So that's fantastic. Job complete. On the other hand, suppose these were like this. So we do the same thing. We kill lots of grays, then we go off and have a cup of tea. Dynamics goes like this, and now you cross the red line first instead of the gray line, and now red decreases, and now you get the gray squirrels. So what this tells us is the same manipulation has given you two different results, and if you're in this case, the only way you can get extinction is if you change these lines. And the only way you can change these lines is if you change the right-hand side of these equations. And that means either changing the parameter values or bringing something else into these lines. And that's precisely the idea that was used. Oh, and then actually if you put diffusion into the system, you get something called traveling waves. So here's a simulation done by a student of mine, Johannes Schluter, from a long time ago. These are the gray squirrels, and they're at 0. And then what's happened is we've put some gray squirrels here, here, and here. And then here's the red squirrels, and they're all at 1. So we've non-dimensionalized everything to lie between 0 and 1. And then if we run this, what you will see is a wave of invasion. and the gray squirrels take over. So now the gray squirrels are all at 1 and the red squirrels are at 0. So we get something called traveling waves. I'll come back to them a little later on. So basically this was looking at what happens if you look locally at the behavior, at the interaction between the two species, and then that other thing was what happens if you look globally. Okay, and so yesterday during the football, somebody was saying they didn't understand what these triangles were, and so this is where you put the upside down triangle. So you have dg by dt equals del squared g plus the function, and that's the diffusion operator. Cells move down the gradient, the g's move down the gradient. And these ideas were the ideas that have been used by a colleague of mine, Bob Gatenby, to look at cancer cell invasion. So he asked the question, so Bob is a cancer radiologist who a few years ago moved to the Tampa to the Moffitt Cancer Center because they wanted to develop a mathematical oncology unit. So he co-directs that mathematical oncology unit with the mathematician Sandy Anderson. So Bob had this idea, let's look at how tumor cells spread through the body as an invasive species. Let's think of it as an invasive species problem. And what we know is that in many cases, tumor cells are glycolytic even when there's enough oxygen. That's called the Warburg effect, been known since the 1930s. Now, why is that a strange thing? It's a strange thing because the glycolytic pathway produces energy at an order of magnitude lower than aerobic metabolism. Now, it's not as bad as that because the glycolytic pathway, you can achieve that pathway quicker than the aerobic metabolism. So it's not quite as bad as this. But still, what he was wondering was how come you've got this invasive species which is generating energy less efficiently than the host species, and yet the invasive species wins. So one thing he thought about was that a byproduct, actually this was for a conference a few weeks ago, so then this person's obviously not at this conference, but this was a few weeks ago. You can see how old this work is. It's almost 20 years old. So the byproduct of the glycolytic pathway is lactic acid that lowers the extracellular pH and is toxic to normal cells. It's also toxic to tumor cells, but not as toxic. So in other words, what this is doing is this is changing the right-hand side of the equation. So he wrote down this model. He and Galinsky wrote down this model. So N1 is normal cell density, N2 is tumor cell density, L is lactic acid, T is time, X is space, so we're just looking at it in one dimension, and everything else is a positive constant. So the original model was the same as the Lotka-Volterra model of the squirrels. It had the interaction terms in, but then they just focused on this simplification for the rest of their paper, but you can sort of imagine that this also has the interaction terms in it. And then what happens is the tumor cells produce lactic acid degrades, diffuses, and the diffusion coefficient be normalized to 1, and this changes the right-hand side of this equation by bringing in a death term. And then they also said that normal cells don't move, which is a fair assumption. Tumor cells move when there's space. So this equation here, if N1 was equal to K1, this would be 0, so there'd be no movement. And if N1 is less than K1, there is movement. And so again, this is very phenomenological, again, we can ask the question, how should we properly model cells trying to move through a crowded environment? So that would be an issue there. We could spend the whole conference discussing that. How does the cell, normally when we write down diffusion equation, we're sort of assuming we don't have a crowded environment. As soon as you have a crowded environment, how does the cell move around? That's an issue that people are working at the moment. But this is just simple, take the simplest possible thing and see what happens. So then what they did, they normalized everything. So everything lies between 0 and 1. And then they said, suppose everywhere as a function of x, we've got normal cells everywhere, and we introduce some tumor cells. What happens? And they found this model gave two behaviors. Both involved invasions. So basically, N2 grew up to its carrying capacity and invaded. And in one case, you've got coexistence behind the front. So here, this is the normal cells, N1. So they've gone down to 0.5. Here, the tumor in the black. And here is the acid, extracellular acid. And intuitively, you can see what's going on here. If you look at these equations, N2 produces L, L decreases N1, that creates a space, and the cells move into the space. Yep?
What has been assumed is, also first of all, I assume these n numbers are at cell densities on a very long time scale, so proliferation is also kind of a movement.
Yeah.
And it is assumed that the normal cell type, the healthy one, does not move at all. I find this approximation pretty strong,
Yeah.
because every tissue must have an ability to repair. Otherwise,
Yeah.
we'd never live.
No, that's right. Yeah.
So if one put in there a small mobility of the normal cells, would this change the behavior qualitatively, or would it be only a renormalization of this N2 term?
Well, that's a very good point, because this hasn't built into it the sort of like a wound healing response, which is what would happen in response to that. And so that's a good question, that what would happen if you put the diffusion term into that? How would that change things? Yep. Yeah. And so I haven't done that, I don't know the answer, but that would be the next thing. That's the rocking back and forth between experiment and theory. So the idea here, start with the simplest thing, see how far that gets us, and then there will be experiments where this doesn't agree with the theory, and then that might suggest, well, maybe the fact that we didn't put the diffusion in there is important. So then iterate back and forth between that. It's exactly the sort of thing one would do. Yep.
So invasion is not really in one dimension. So what would the geometric effects be predicted by this model if you allowed it to happen in 2 or 3 dimensions? Would you get the kind of fingering-like invasion that you see, or is it always some kind of a substrate-wide front, the way the 1D predicts?
Well, I think probably, I don't know, because again, this is really, as far as I know, only been simulated in 1D. So that's actually another good question. I would think probably if you did it in 2D, you would still get a wave, but then if you had heterogeneity in the system, maybe that could break. Like, because, you know, if, for example, the D2 was spatially heterogeneous or something, you might get the fingering. But I don't know, because I don't think anyone's ever done this in 2D. And Thomas, yep.
And just to come back to Rudolph's question, I think if you actually added diffusion to the first equation, you'd kill off your normal cells quicker, because one, they'll be moving into the population where it's producing lactic acid, so it's killing them off. And secondly, by allowing them to move, you're creating space for the tumor cells to invade into.
Yeah.
So actually having them move hurts you.
Yeah. And of course, you've got to remember that, you know, this is a very much a simple case of looking at normal cells and tumor cells. It's not bringing into account things like the immune response and macrophages and all those sorts of things that would be coming in.
A question? So this actually raises the question that you talked about earlier. So what is the next point in the cycle in trying to see what value this equation has?
Yeah, so first of all, if we see what the results are and then the testing of the prediction before we, so then we would need to see what is the next part of the cycle. So in fact, in this one, as far as I know, I mean, this isn't my work, there is now a lot more detail being gone into this by Sandy Anderson, has a lot more biological realism into this after this first round of talks. But if we go back to see the behavior, so here we have then an invasion of the wave front. In fact, a very simple thing that comes out of this is that this can only give rise to two behaviors, and both are invasion. And that's actually because in the simplified version of the model, they didn't allow for the normal cells to inhibit the tumor cells through, say, an immune response. So the next cycle is to actually put the proper Lotka-Volterra in, and then you do get behaviors like clearance of the tumor, pinning of the tumor, as well as the invasion. And now this second invasion type of behavior you get depends on the parameter values. The second system, and now you notice two differences between these two. One difference is the normal cells have gone to extinction behind the front. The second difference is that you have here, there are no cells. So that was a prediction. They then thought, I must go and see if that's true. And they studied head and neck cancers, and they found this. In 1/3 of them, this thing here. Now, this is a controversial picture. This is a gap between the two cell types. This is a controversial picture because a lot of people will say this is an artifact of the process that was done to get this picture. So this is something that, if you're from mathematical training, you need to bear in mind that just because it's in a paper and just because it's a picture of an experiment doesn't mean it's real. Okay? One thing I've noticed is mathematicians seem to have this great belief, great respect that everything that's done in biology is correct. And actually, biologists have this great respect of mathematicians, that the mathematicians are super people, can solve any problem. And it's really nice having that mutual respect, but I think sometimes the field might advance more if there's a bit of disrespect. Okay? And in fact, I think this is one thing that's been really good in this conference, where people have really been asking questions. What about this? What about that? Rather than saying, you're so clever. The process by which you get this picture actually can make this bigger than it is. So they actually also had pictures where they did a different process, which didn't involve fixing, and then you got a gap, but the gap was much less dramatic. So that's one thing also to keep in mind. Sometimes experimental result might be an artifact of how the result was got. That's why it's actually very important to talk with biologists to find out exactly what they did. And then here are what they did also was they then took cuts through the tumor to measure the pH, and here are the pHs, and this is the tumor, and this is normal, and so what you have then is the gradient of acid that looks like this. Okay.
Do you happen to know the scale?
No, and the scale here, I mean, you're talking about a few points of pH. Yeah. The important thing is this is very qualitative because the parameters are not parameter values that are measured from the experiment. Okay, so obviously this then, so we've tested the hypothesis. Remember, the hypothesis was that the tumor cells, by producing acid, changed the environment and allowed the tumor cells to invade. That was the hypothesis. So write down a mathematical model, that hypothesis holds, but gives you something you hadn't thought about, which was that in some cases it'd be a gap. And then the gap was found. Okay? So now this means, well, maybe this is useful to take this a little bit further. Let's add bicarbonate and see if we can buffer the acid and stop the cells from invading. So that's what they did. So they took a mouse, put tumor into the mouse. This is on the window chamber at the back. Then one set of mice, the drinking water was full of bicarbonate. Other set of mice, drinking water was just normal drinking water. These are tumor cells that metastasize to the lung. And then they looked to see, was there any difference between the two? And what they found here was untreated mice. You have a statistical difference, this is number of metastases, between untreated and treated. Okay? So now the question is, what is, as Hans pointed out, before we go on to the buts, what is the next process in the cycle? As quite rightly pointed out, there's no wound healing response. Should that go in? Another thing is that tissue is not just cells, there's matrix. So the tumor cells need to secrete proteases to break down the matrix. And in fact, the secretion of the proteases can actually be upregulated by the acid. And so people like Steve Webb, for example, in Liverpool, he's done a model of that. So there's now a whole range of different directions in which you can do the next iteration. And the question is, which direction do you go? And that depends on maybe the experimentalist you're working with. Okay, if you're a mathematician, the next direction you go is you prove existence and uniqueness theorems, and you change the right-hand sides so that you can do existence and uniqueness. If you're an experimentalist, you will say, well, this is the only experiment I can do. So then maybe that could be a way that you decide which direction to move into. Yep.
There's another thing that always bugs me. It's in nonlinear systems you can get the right answer for very wrong reasons.
Yep.
Do you know a systematic or a reasonable way to tackle that one?
I think that, well, actually I don't is the answer to that. I think just by going into more and more detail, by really sort of testing the model in more detail, doing more perturbations to the model and comparing that with the theory. I think that would be one way of doing it. But what, yep.
Philip, I think you can make a stronger defense than that, which is that experimentalists also get the right answer for the wrong reasons.
Right.
How many knockout experiments have you done of a molecule that is key to process X, which then has no effect, has no phenotype?
Yeah.
So,
Sure. Yeah.
so at least in the at least in the simulation, you know all. I think that's something maybe to have emphasized even more at the beginning, which is that in a simulation you only have what you put in, and so in a simulation you know what mechanisms you have, whereas in an experiment you have a picture of a pathway which may or may not have anything to do with the actual interactions in the cell. And so the one big advantage here is that you know what you're working with.
Sure.
And so that's really
Yeah,
critical.
that's right. Yeah, and you can invalidate it. Yeah.
Yeah, but against that comment, one could also say that there's probably a relatively large equivalence class of models that will predict the
Yeah.
same thing.
Yeah.
So that in some cases in the modeling, you end up doing little more than curve fitting in the Banach space. So I think one has to keep in mind that there's no uniqueness about a model that will predict what you're observing. There's a variety of processes. Just as a simple example, in this context, if you don't have a diffusion process for the cell movement, but something more complicated, you end up with a hyperbolic equation and you will see the same sort of behavior.
Yeah.
So how do you distinguish between the two?
That's right. Thomas?
I think what Hans has just done is sort of given us a defense for theory's sake, because as you showed that the model for Gauge-Me's model is you only have these two distinct modes of interaction, and so you could change your parameter value by one, you'll get the same interaction. Change it by one, interaction. So by looking for the bifurcation structure, which is a very theoretical thing to do, it may not help the experimentalist, but you can see everything you can get from the theory.
Yeah.
And it's a really nice way of categorizing. So as Hans said, you can have all these different models which do the same thing, but we know because of the theory. And so when biologists quite often say, well, we know that, like, yes, you do, but we can say why we know that.
Yeah, that's right. Yeah. So if you move on, there's another issue about these models, and that is that these traveling waves, these are called pooled fronts. So their behavior from a mathematical point of view is determined by what happens far ahead of the front. You can do that mathematically rigorously. It all makes sense. It's all fantastic, nice theorems, except if you want to apply it, because far ahead of the front is the one place we know for certain this model is not right. Because far ahead of the front, there are only a few cells. Stochasticity is important. You can't represent it as deterministic. Okay. So this boils down to this other thing that's been mentioned again in the past. Behind the front, we can write down PDEs. This macro scale, we know how to deal with macro scale. Ahead of the front, the micro scale, we can do individual based simulations. We can do stochasticity. How do you match them? The mesoscale. Now, people say the devil's in the detail. I say the devil's in the mesoscale. How do you match what goes on at the front with the back? That is an open question. There's another but, and that is these models make the assumption that the system and the interactions are well mixed. And we write down the law of mass action, which says that the rate of reaction of A with B is basically A multiplied by B. So this has worked with colleagues, go back here, Debbie Markham, Matt Simpson, Eamon Gaffney, and Ruth Baker. So what we did here was we took two species, A and B, we had them interact with each other in a Lotka-Volterra type interaction, and we had that A would win. Okay? So if we look at black, black is a result of simulation. There's A in the full line, and here's B in the dotted line. It goes to 0. We now do the Lotka-Volterra. Okay? In the purple lines. And here's one species goes to 1, the other species goes to 0. It's the wrong species. The model got the wrong answer, totally the wrong answer. And the reason for that is their spatial correlations. In a biological system, if you're A, chances are nearby you are also A, because you divided and maybe didn't move very much. And how we did this simulation to make absolutely sure that the Lotka-Volterra would give the wrong answer is that we had these species divide a lot and not move very far. And then you have to take spatial correlations into account, and then if you take spatial correlations into account, you do get the right answer. but I'm happy to go into that in detail, but let's just move on. So not only do we see that there are issues about whether, what else should you include in the model, there are also technical issues. Now, the new people in the audience are probably sitting there thinking, one thing that's becoming very clear about this talk is that we know nothing. Or in fact, what's become very clear is that Philip knows nothing. But this is the really exciting bit. We know so little. And you guys are so young. You've got 40 years in which to discover all of this stuff. It's really exciting. You can make fundamental advances. You started your doctorates last week. By next week, you can make fundamental advances in the field.
Philip, may I give one more add-on to
Yep.
that, because it's such an important point. You talk about spatial correlations, which are critical. Even in non-spatial models, maybe especially in non-spatial models, we tend to use reaction kinetic equations, which are deterministic. And so you take a simple circuit like a latch, and in a deterministic model, it latches, and that's it. But when you add stochasticity, there's always going to be some probability of the latch reversing. And so what you'll get instead of a one-time switch is something that switches continuously depending on the noise in the problem. And so when you look at a pathway diagram, and you think about that in terms of ODEs, you may get qualitatively, not just quantitatively, but qualitatively wrong behavior.
Yeah, that's right.
And you've also mentioned here, more at the tissue level, the issue of segregation and mixing, but also within a cell, cells are not stirred reactors.
Yeah, that's right.
So
Yeah, with inside the cell.
matters a lot whether two components that can react actually come into contact with each other.
That's right.
And they don't necessarily do it just because they're inside the same cell.
Yeah. And I think this is where you have to be very careful. So these pathway diagrams are like Frankenstein monsters, because you got each interaction from a different experiment, and then you put them all in the same diagram. But as James quite rightly points out, maybe in real life these things are never near each other in the cell you're looking at. So you have to be careful about that. So the last thing I want to talk about is another invasion, but this is a normal invasion, normal development. This is work with colleagues Paul Kless and Rebecca McLennan in the Stowers Medical Research Institute, and colleagues Louise Dyson, Ruth Baker, and Linus Schumacher. And the general idea here is that in early development, you have cells that come out of the neural crest, and they have to move along and need to find their place in the branchial arches. And different streams go to different places. So from an abstract point of view, here's the question. Cells are coming out into a domain. That domain is growing. How do the cells reach the end of that domain? Seems a very, very simple question. We're going to talk about neural crest, and I don't know about the other invasions, but in neural crest, there's no cell division that happens during this process. This is important because if it doesn't happen properly, bad things happen, and may be important in cancer, we don't know. Now here's another thing that I think is very important. These are just to remind me to say to you that there have been huge advances in imaging. And now for the very first time, I think in mathematical biology and dynamical systems, we have the opportunity to really test our models. Because beforehand, all we had were snapshots. And if you want to test a model that describes how things change in space and time, having a snapshot is useless. But now they can do things like they can follow a cell. These are different tracks of cells. They can measure how the domain grows. They can do all sorts of things. And I think this is hugely exciting for us now because we can really try to validate our models. So the first hypothesis to test is the following. It's known that VEGF is produced by the tissue. It's known that cells eat up VEGF and move up the gradient of VEGF. So there's an obvious first model. The cells come out, they start to eat the VEGF, that creates a gradient, they move up that gradient, they're continually eating the VEGF, and so they reach the end. They reinforce the gradient. So let's test that hypothesis. So again, we write down a diffusion equation for the VEGF. The cells we model as points that use up the VEGF, and that's just this thing here. VEGF is produced, and this is dilution due to the fact that the domain is growing. Here, we just take this function that fits the curve, the growth curve that Paul gave us. And the cells we model, they come out into the domain, they randomly stick a hand out in a random direction, they measure the chemoattractant concentration there, compare it with the local concentration, and make a decision as to whether or not to move. Okay? And so now let's see what happens. So this is the domain, and it's all red, which means there's VEGF everywhere. The cells are coming in, they're creating this gradient, and then very soon it will all go wrong because there's no gradient here for these cells.
So what are the rainbow colors?
Oh, these are the VEGF. So concentration of VEGF, yep. And so now what's happening is these cells have got stuck because the gradient is only at the front.
But is there a relationship between the growth rate of the domain and the rate at which they are invading that domain?
So at the moment, all we do is we just have the cells coming into the domain when there's space available. Yeah. So then we said, so what Paul had noticed was sometimes two of the cells seem to be stuck together. So then we said, well, let's make the assumption that there are two cell types. Cells at the front that respond chemotactically, cells at the back that stick to cells at the front. But the way we did this in the model was we said that a cell at the back randomly sticks its hand out. If it grabs hold of a cell at the front, it takes the velocity of that cell. So that means it could have stuck to the cell, or the cell at the front could have created a nice track, so the cell at the back follows that track, or the cell at the front has laid down some signal, the cell at the back follows that. The model can't tell the difference between those. So now let's do the simulation for that one. So it starts off as before. You've got cells at the front, they're coming out, and they're responding to the gradient, and now the gradient disappears, and now you've got these followers coming out.
What are the little spikes on the cells?
Oh, those are just randomly where they're supposed to be Philopodia randomly where they're trying to find a position.
Sorry, Philip, so how do you distinguish the cells that are followers from the leaders?
So at the moment, and so we'll come back to that in a few minutes, at the moment we're just saying it's hardwired.
Okay, so
yeah.
Because you certainly don't want your leader cell to reach back, find the cell that's not moving, and then stop.
Yeah, but we'll come back to that because we'll see that that is not as simple as that. Okay, so that was in case the videos didn't work. So what we have then, a prediction of the model is cells at the front and cells at the back are different to each other. So they did, well, the first prediction is that the simple model of chemoattractic gradient and a single cell type doesn't work. You need to have at least two cell types. They then showed there were at least two cell types. In particular, they showed that the cells at the front upregulate things like proteases that are needed to degrade the matrix, and various cadherins, etc. And the cells at the back upregulate different cadherins. Some cadherins are actually to do with the phenotype of stickiness. So then we did some experiments, and what we found was sometimes the model is correct, put that in inverted commas because model isn't really correct. Sometimes the model is incorrect, and sometimes the experiment is incorrect. So here's the first experiment. What happens if you remove the trailing cells? So we meet with experimentalists, we design the experiments with them, they go and do the experiment, we do the theory. We don't talk to each other during that time, just in case we accidentally let each other know what we're thinking. So you remove the cells at the back. The model prediction is that you get the cells strung out, and that's what happens in the experiment. This is the wild type, and this is the experiment. So the next, what happens if you take lead cells from a donor, stick them to the back of the host? And so they showed us this picture here. So this is the host, and this is the transplanted cells. And we said, oh, that's really terrible because our model predicts that nothing will happen. And they said, no, that's really fantastic because in most of our experiments, nothing happened. But we didn't want to show you a blank slide, so we found an experiment where something did happen and we showed you that. So there's an example where the model was correct, the experiment was wrong. What happens if you take cells at the back and stick them to the front? Now in this case, the model is so wrong, I won't even tell you what the prediction is. Model is wrong because what we see is the cells at the back, they start to invade. And so what we realized then, model was wrong, what we realized then was the cells at the back switched their phenotype and became front leader cells. So now it's time to get up onto the virtuous cycle again, do the rocking back and forth, whether you want to cycle or rock, up to you. So we've made predictions, we've tested it, we've refined the model, and we predict. See, refine, when people say refine the model, that's really double speak for the model was wrong, so we changed the model. Okay, but we say we refined the model. Okay, so we refined the model. Okay, makes it sound more grown up. Then what really happens is you say the model is wrong and you stamp your feet. So now there's two things. One thing is I never believed that VEGF was important. So I kept saying to Paul, do the experiment of stick VEGF along the trail and see what happens. Easy for me to say, very difficult for experimentalists to do. So eventually they've done that, and I won't go into the details of that because I don't have any time. Instead, I want to look at this question they asked us. So they asked us the question, how many leaders do you need for this to work? And of course, because we've got a model, we can make as many leaders as we want. As few leaders as we want, we can do what we want to do. So different graduate students, so different color scheme. So now this color here is leaders, and this color here is followers. So here's a situation where everybody's a leader, okay? And that's what we get. Here's a situation where there are very, very few leaders and most are followers. And there's two things to notice here. One thing is, it doesn't matter how many leaders you have, you always reach the end. Second thing to notice, this thing here, this color is the average density. And what you can see here is that here, lots of cells are moving in. Density is very high. Here, not so many cells are moving in. So this is not a good invasion. This is a very good invasion. So the prediction of the model is you only need a few leader cells. So what they did then was they expressed HAN2, and HAN2 is something that upregulates the leader phenotype. And You get to the end, like the model predicts, but you've got less density, like the model predicts. Then what they did was they expressed this thing here, and this thing here downregulates the leader phenotype. So what happens? Again, you reach the end, but you've got a better invasion. Again, as the model predicted. So what comes out of this is if you have too many leaders, it doesn't work. And that's something we already knew. Well-known saying, too many chiefs, an insufficient number of indigenous persons. And this paper was just published a few weeks ago. So there we have that rocking back and forth. Predict, refine, predict. Predict, test, refine, predict. Okay? Now here's a question that I think is quite a nice abstract question. You might think, 5 minutes, okay, just about finished. You might think, actually, it's so easy. What's the big deal about this? Just create a gradient, have cells move up the gradient, and they get to where they want. Now, it turns out, in fact, Roberto Mayor has done a lot of work on this at UCL for other neural crests, and he's come up with the same thing, that not only must you have some sort of gradient that's allowing the cells to move in a direction, you have to have some interaction going on to get everything to move. And the same thing happens in angiogenesis. In angiogenesis, you have cells, in tumor angiogenesis, cells move up a gradient of VEGF. And then there's a snail trail model that says that as the tips move, they leave behind them, the cells behind them divide. And so that's how the trailing cells follow. Or there's an alternative, which could be that the cells at the back push the cells at the front. Either way, you need at least two mechanisms. You need to have some global signal that tells you the direction, but somehow something else happening within the cells to get them to move. So it seems to me that this is something with at an abstract level, mathematically, one might be able to tackle. Write down, say, if I want a population and I want to move it from one place to another over quite a long domain, what are the minimum requirements I need? And then let's see, of those minimum requirements, which ones do biology pick? So I'll end there. A few weeks ago, I was at UCL at a conference, and they had an artist in residence. And so while we were giving the talk, the artist in residence sat over there, and he drew cartoons talk. And then at the end of the day, so this artist, not a mathematician, not a biologist, went to art school, did art. At the end of the talk, this guy got up, he went through every single talk, and he recounted the key elements of every single talk. It was the most extraordinary thing I've seen. So here is the neural crest, and he remembered that it was chick because the lead cell has got a beak. The VEGF are croissant, they're moving along, and here's a cell needs to decide, is it going to be a leader or a follower? Here, glycolytic, the N2, firing lactic acid, and here is the gap, and here's the normal cells, and they're firing bicarbonate. Now, when I was a little boy, and I would get sick and I'd get given tablets, I used to imagine in my mind that out of those tablets came a big army, and that army had a bloody battle with the germs. And it turns out that's actually what happens. It's so fantastic. And also in that talk, which I haven't mentioned in this talk, we looked at how you could apply this to humans, and what we found was that it wouldn't quite work in humans, so we suggested, well, just give more bicarbonate, and then they said, oh, that's poisonous, and that's what's happening here. There's a biologist saying, oh, this is toxic if you put too much in, and this is a mouse, experiments done in the mouse. So by writing it, by putting all those key things in, he was able to, And what he actually does is he trains people, because we all assume that just because we learned by somebody standing up and talking at us and we make notes, we assume everybody learns that way. That's not the case. Some people learn this way. So he trains people to do this.
It's a wonderful drawing, but what's the pink head in the back?
The pink head in the back, I don't know what that is. Well, yeah, that's the neural crest, but that's a good question. Why didn't he draw a chicken head for that?
Oh, I guess that's Gatenby.
Yeah, that's Gatenby there, Robert Gatenby.
That's you.
And that's me. Different shirt, but still me. Okay, and so the last thing is just an advertisement. A few weeks ago we published this thing, What Has Mathematics Done for Biology?
They did the other way around. What is biology done for mathematics?
Keeps us busy, yeah. So I'll end there. And one thing that I want to try and get across is to the younger people in the audience is that you should sort of see, and I think this has been very good, this workshop, is that, in fact, one of my postdocs one time, having been to a workshop and been to a conference, described the difference between a workshop and a conference is that you go to a conference, it's like you're on your first date. So you're well behaved and you tell everything, oh, this is fantastic and all this sort of thing. A workshop is once you're married and you let the real honesty come out, okay? And that's what this workshop has been. And I hope that you then see that there are so many places in which you can make contributions, not only in the biology, also in the modeling, also in the mathematics. And there's one thing for sure, is that over the next 40 years of your career, you will not ever have a boring moment. There's so much for you to do. So I'll end there and thank you for your attention.
We've got a lot of questions en route. No, back here, just a few because we're almost at coffee time.
In your VEGF model, where does you, what's the source for the VEGF? Is it
So what we say is just the tissue. Yeah, we're just making the assumption that the tissue is producing the overlying tissue.
continuously producing the same amount of VEGF?
No, well, in the model we have it's producing up to a certain level. So it's not continuously producing it. So it produces until it reaches a certain level, and then it won't produce beyond that. So we have a logistic growth of that. Yeah.
But it would continuously produce up to this level.
Yeah, up to and then it would stop, and then as it becomes degraded, it produces back up to that level. Yeah.
Quick one?
So, Philip, your comment about the bicarb is a great one, and it's maybe worth, especially for the students, to explain why the bicarbonate doesn't work. Mice don't regulate blood pH very closely,
right. Yeah.
and therefore, if you give them bicarbonate, the blood pH shifts very rapidly.
Yeah.
Human kidneys are extremely efficient at regulating blood pH, and therefore, you can give humans an enormous amount of bicarbonate. The blood pH doesn't shift, it just puts a load on the kidneys, which eventually fail. And so there is a difference in kidney function that means that the experiment doesn't work.
Yeah, so in fact, that's one of the conclusions of the model, is that people with bad kidneys would do better for precisely that reason. Yeah.
And I think that actually brings us back to the front where Philip mentioned that a model system, a mouse might not be a model for a human.
Yeah, that's right. Yeah.
Well, I think we'd better stop there and leave the rest of the questions for the coffee break. Let's thank Philip again.